Okay, let's go ahead and start. So last topic we want to deal with in terms of bacterial diversity in this module are microbial mats and biofilms. So we'll start with microbial mats first because these are actually some of the most ancient forms of microbial communities known. There is fossilized evidence of microbial mats existing more than 3.5 billion years ago. So these are assemblages of microorganisms that typically occur at the in the environment and typically at the interface between like water and a solid substrate. So these microbial mats, while they were probably the most dominant forms of life for millions or billions of years, now are relatively restricted in terms of their environment. You find them in association with salt marshes, you find them in association with intertidal zones, shallow areas of the ocean where they don't have a lot of competition. As it turns out, as multicellular organisms have evolved and evolved the ability to burrow down, so things like clams and things like that, they've actually inhibited the ability of microbial mats to live in a lot of areas because parts of these microbes or these mats require anaerobic environments. So typically these microbial mats are multi-species communities, and what distinguishes them from the next topic that we'll talk about, biofilms, is that microbial mats almost always have, if not always have, some type of a primary producer. Oftentimes it's a photosynthetic organism, but it's not always photosynthesis, but there's almost always some type of an autotrophic organism that can take inorganic carbon and fix it into an organically usable carbon form that other members of the species, microbial species that are living in that mat can then use. So these are very self-contained ecosystems using oftentimes sunlight, but a lot of times things like hydrogen sulfide. So they're still associated with things associated sometimes with hydrothermal vents. So you can find them in particular areas like in Yellowstone National Park. This is the Grand Prismatic Pool from Yellowstone. And so there are microbial mats that are living here on the edge. So in salt marshes today, microbial mats are still an important ecosystem to consider, and they change over time. So in terms of the dynamics of the structure of this. So in salt marsh microbial masses, a bacteria that we talked about earlier in this semester, Beggiatoa. This is the bacterium that was first discovered to be able to oxidize hydrogen sulfide, that colorless, rotten egg smelling gas that's poisonous to us. This bacteria can oxidize this in a chemolithotrophic sort of a way. And this microbe exists in association with a photosynthetic bacterium, Oscillatoria, which is a member of the cyanobacteria. So these two species, along with other species, exist in these mats. But then as the daylight changes and oxygen levels change, these microbes actually migrate to different regions within that mat. So the Beggiatoa exists at this interface between the water column and an anaerobic environment via three taxes. It avoids light, it tries to get to hydrogen sulfide, and it tries to avoid oxygen because Beggiatoa doesn't like oxygen. So during the daylight hours, when the microbial mat is exposed to light, this motile bacterium, this chemolithotrophic bacterium, migrates inside the mat. Because what's going on during sunlight is the Oscillatoria is accomplishing photosynthesis and producing oxygen. Beggiatoa doesn't like that, so it chemotactically moves away from that oxygen concentration. Then at night, when Oscillatoria can no longer be producing oxygen, it can move out. But it also keeps it in a very narrow range of optimum for hydrogen sulfide, which it's using as its electron source. So these microbial mats are actually pretty fascinating for a large group of people that study environmental microbiology. But in reality, I think probably for you and I shouldn't assume that you're not interested in environmental microbiology, less so than I am. But I think biofilms, which are related to microbial mats, are much better studied. And so we know a lot more about them. But most of what we know about microbial mats and biofilms actually comes from studies on environmental microbiology. So moving on to talk about biofilms, these are often, so probably the primary difference between biofilms and microbial mats is that biofilms typically do not have a primary producer. So there's very seldom, if ever, photosynthesis going on. There might be an autotrophic bacteria in there, but for the most part, they don't have a primary producer. So they tend to be organisms that live in environments where there's flowing water, usually water. We have biofilms in our mouths right now. We'll talk about those. That's dental plaque. But in these flowing environments, this ability of bacteria to live within a community made up of excreted extracellular polysaccharides, very similar, or if not identical, to the same polysaccharides that bacteria produce to create capsules that we talked about. Now, bacteria living collaboratively, all producing capsular material, end up producing what's known as a biofilm. And these biofilms that have the bacteria living within them are actually highly structured environments. They're not just these slimy piles of goo. They actually have channels in them to allow water flow so that nutrients can be brought in and waste products can be taken out. So dental plaque is probably one of the most highly studied forms of biofilms. And for the most part, most people in the biomedical field really felt that dental plaque was perhaps the only area in which, of human health, in which biofilms actually had any type of an impact. So for the most part, most of it was studied. So here's a tooth on the bottom here, here's the enamel, and so you have microbes attached to that. And if you're not doing proper oral hygiene, the plaque will develop and these biofilms will be produced that have very huge numbers of species, and it's amazing the bacterial diversity that exists within our mouths and within dental plaque. But actually, they will form water channels through there to allow food and water as we eat to get in order to feed these microbes. There's a big advantage to microbes living in biofilms like this because, kind of like capsules, these gelatinous materials protect the bacteria from toxins and antibiotics. It becomes very difficult to treat microbes in a biofilm like this with antibiotics. I'll show you a video in a little while because one of the main reasons we know now for the failure of artificial joints, so people with hip replacements or joint replacements, or things put into their spinal cord. One of the major reasons for the failure of those is because bacteria get associated with those artificial plastics and form a biofilm. And then that biofilm is so protective that you can't get antibiotics in to kill the bacteria. So oftentimes what happens is that these prosthetics have to be replaced. So biofilms are very important medically, certainly in the case of replaced heart valves, for instance, and also joints. So they protect the microbes from antibiotics. They also allows for very efficient quorum sensing. So the secretion of peptides or homoserine lactones occurring within this extracellular polysaccharide tends to not diffuse as rapidly. So it's not lost to the flow of, in this case, saliva, or it could be in a stream. So the bacteria living in the community can sense a lot better their own senses, and so they can change their gene expression a lot more effectively. It's also great for genetic exchange. So bacteria, as they die and release DNA into this environment, other microbes can pick up that DNA. So evolution, the stuff of evolution, these genetic mutations in some bacteria are actually more easily picked up because of bacteria that die within these biofilms. So the biofilms allow these bacteria to adhere as well. So it protects them in high flow environments from being washed away. And it also allows them to control nutrient diffusion. So if you have a microbe in a stream, for instance, that's attached to a leaf that it can degrade, if it forms a biofilm, then as those bacteria collectively, under the protection of quorum sensing, degrade that leaf, as those amino acids and sugars are released, they don't just simply get washed away into the stream. They are protected by that film, the biofilm. And for years, people thought that biofilms had virtually nothing to do with human health. But now, as people have developed better and better tools to study microbial biofilms, we realize that a lot of human diseases, including tuberculosis, staph infections, kidney stones actually begin or have a portion of their pathogenesis that has to do with biofilms. And cholera, the organism that causes epidemic cholera, actually exists as a biofilm within the human body during infection. So there are significant causes, as I mentioned, of failure of medical implants, stents, joints, and heart valves. They're also a major problem for fouling. So this is a pipe that's been used for several years for water flowing through it. And then what you see here is a microbial biofilm that has built up within that pipe over time. So the microbes are extruding this, they're living in here just fine, but now the flow of fluid through this. So this is a major problem for people trying to maintain pipelines and things like that. It's also a problem we're becoming more and more aware in human health. So there's a bacteria, and we worked with this a little bit in the laboratory, Pseudomonas aeruginosa. Pseudomonas aeruginosa is an opportunistic pathogen. So for you or I, relatively healthy people, it's not so much of a problem. But for burn patients, it's a tremendous problem. For cystic fibrosis patients, it's a tremendous problem. So what we know now is that these are some pictures I stole from a study that came out 5 or 6 years ago, studying Pseudomonas. If you look at it, so this is Pseudomonas being grown on a glass cover slip in the laboratory and stained for DNA. So the bright spots here, like one shown here, is the bacterium Pseudomonas. But you see the diffuse amount of DNA that's in here. That's extracellular DNA. So these bacteria, as they're dying, are actually using the genetic material of their dead brethren to help structure this biofilm. If you look at a sagittal view, so they've false colored the cover slip blue. So now you're looking not on the cover slip flat, but looking at it on its side, you can see the thickness to this. And again, you can see the individual bacteria staining bright green. This has actually been overexposed. And you can see the diffuse DNA or extracellular DNA. So these biofilms are not simply capsular material. They're not simply polysaccharides. They actually are they're more complex than that. They have genetic material within them as well. Now, one of the reasons why it's important to understand these kind of things is that this is a man we'll use for an example, or I use for an example, Anthony Brennan. He's a material science engineer professor at University of Florida. So he came up with this material, which is now widely used in hospitals. The material is called Sharklet. It's a plastic material that he designed to be resistant to colonization by bacteria, so biofilms don't form on this type of film. He actually was inspired for this because of his interest in marine science. He's not a marine scientist, but he realized that sharks, unlike many organisms in the ocean or anything in the ocean, were particularly resistant to fouling. You know, shark skins are very smooth. They don't develop biofilms very often. They don't get barnacles attached to them. Whales get barnacles attached to them. Ships get barnacles attached to them. And so there's a major, so his research really was trying to, for the Navy, for the most part, was trying to figure out how to prevent ships and submarines from acquiring bacterial biofilms, which could then potentially lead to barnacle fouling and things like that. So he noticed that sharks, because they have these denticles, here's what shark skin looks like. He had actually come up with a mathematical model that predicted that if you made something very similar to that with these little ravines and ridges on here, that perhaps that would inhibit microbes from binding. And so what's shown here is his sharklet on the right over time, cultivated with bacteria, as opposed to just a smooth plastic, the same material. And there's something about these ridges resembling the shark skin denticles, which actually inhibits bacterial biofilm from forming, and in fact, it does better than that. It really, for the longest period of time, it prevents bacteria from even colonizing very significantly at all. So those of you who go into the medical field, you know, note when you're in the hospitals, this is a relatively recent advance, but I know now that he actually has a company. I think he has maybe since left University of Florida because maybe he can make more money with this, but he certainly at least has a company called Sharklet that makes this kind of plastic for hospitals, and it's all based on his knowledge of how bacteria attach and form biofilms. So I'll play a little bit of a video now that's animated that will show you a little bit more about biofilms.
Bacteria enter the bloodstream from various infection routes and evade the immune system of antibiotics. Formation begins with attachment of planktonic bacteria to the surface, preconditioned with proteins from the host immune system. Irreversible attachment to host proteins signals the bacteria to create biofilm and is the first committed step in biofilm growth. The bacteria anchor themselves permanently using cell adhesion structures such as pili and receptors called epitopes. This encourages new bacteria to attach to newly arriving cells, providing adhesion sites and building a protective matrix of the biofilm structure. Cells now communicate by quorum sensing and use small molecules to regulate gene expression within bacteria. The community grows through both cell division and recruitment. The development of this complex biofilm makes the community resistant to the immune response and antibiotics. It may include diverse species of bacteria, yeasts, minerals, and host proteins. Recruited neutrophils result in a cascade of inflammatory cytokines and host proteases. These damage tissue and regulate fluid accumulation next day, providing nutrients. Bacteria now exist in multiple forms and metabolic states. Now a different phenotype, both physically and metabolically, has the survival advantages of diversity and multiple defenses. The life cycle continues as fragments of planktonic bacteria are released to reinfect the human host.
And have to get the credits in there, otherwise YouTube might not let me post this. Yeah, and as I mentioned, humor is pretty scarce in microbiology, but here's a bacterium. Just can't go with the flow anymore, thinking about joining a biofilm. Before we leave biofilms, and actually I'm going to show you a slide and we'll talk later about dental microbiology. But, you know, just in terms of current events, one of the things, you know, you don't think of biofilms as being involved in, but biofilms are critical for those of you who've been paying attention to what's been going on for the past couple of years in Flint, Michigan, where large numbers of children, well, everybody has been exposed to high levels of lead, but lead poisoning is always more of a problem for children, right? It leads to cognitive problems with children developing. Well, so that actually is underpinned by biofilms, because what's happened is that basically in any house in the U.S. built before about 1930, that house, if it's unless it's on a well water supply, if it's getting a water supply from the municipality, probably has a lead pipe bringing water into the house from the main, and then a lot of the pipes are welded with soldered with lead. So why is it that, you know, not more people are getting lead poisoning from their water? And this was the case in Flint, Michigan. People weren't getting lead in their water until somebody decided to save some money. So Flint, Michigan, you probably know this story as well as I do. City administrators decided to save some money, and they stopped buying their municipal water from Detroit. The city of Detroit got their water from Lake Huron. Lake Huron's a relatively pure lake. You know, the water's still processed, but it's a very good source of water. To save money, they started getting their water from the Flint River, which is a little bit more polluted, but more importantly, it's more acidic. What happens is that this acidity in the water, since they didn't condition the water, so you may have heard about this in the news, that if they had only added phosphates or something to adjust the pH from its slightly acidic level, there would have been no problem. Because what the acidity of the Flint River water did was it killed the bacterial biofilms that had built up over years on those lead pipes. So what was protecting the residents of Flint, Michigan, and many other cities throughout the country is the fact that over the years, those lead pipes are now coated. So the water that's coming into their houses is not actually coming in contact with the lead. And this is why, most municipalities recognize the fact, so this was the criminal part, I think, of Flint, Michigan, is realizing that they were not only going to save money by not buying their water from Detroit, they were also going to save money by not conditioning the water. If they'd only conditioned that water for pH, there would have been no problem. That they saved money on that end as well. That's really the criminal part of it, because if you read stories, other cities that have to deal with this very strictly regulate their water pH, and they add things like phosphates or silica-based pH things to protect those biofilms, because there's just a thin film of bacteria protecting those lead pipes from being corroded by that acidic water. So biofilms are actually really important, more than you could ever know. So this is actually, this is one of my lectures that's probably going to make you want to go and brush your teeth again, even after lunch. So this is an assemblage of the microbes that can occur in your mouth. Probably not in any of your mouths right now because your hygiene is probably pretty good. But here's the tooth surface. And so this is kind of an advanced ecosystem that occurs in plaque. So plaque, to some degree, is normal and protective of teeth in the same way that biofilm protected the residents of Flint, Michigan from lead poisoning. There are salivary proteins that are going to deposit on your tooth surface. So imagine coming from your dental hygienist where you've just been scraped and polished and your teeth are as clean as they're ever going to be. Within minutes, probably by the time you leave the office, your teeth are now covered with what's known as an acquired pellicle. These are salivary glycoproteins and such. But they actually serve somewhat to protect the tooth, but they're also there to bind to protective bacteria. So the first bacteria that are going to colonize your newly cleaned teeth from the hygienist's office are going to be streptococci. And these streptococci, although you think of them as pathogens, these are actually protective organisms. So if your dental plaque never develops past this first group of microbes attaching to the salivary pellicle, you're fine. You're probably not going to develop periodontal disease. You're probably not going to develop dental caries or cavities, because these microbes are protective. So as long as you just brush and floss, well, you get rid of these bacteria, they come right back. The problem is if you're not properly maintaining your oral hygiene, then this whole other group of microbes gains access. And they can only gain access if these protective bacteria are there, because you notice these receptors are kind of fitting like lock and key. So certain microbes like this Propionibacterium acnes can bind to Strep gordoni. But if Strep gordoni wasn't there, it wouldn't persist. So this is a really structured assemblage of microbes with early colonizers and then late colonizers. And some of these microbes are seriously bad actors. So I don't know exactly what RED stands for. I tried to look this up several times, but several of these bacteria fit into what's known in the oral microbiology world as the RED complex. These are microbes that are known to be associated with periodontal disease, especially adult periodontal disease. I don't think people still know what causes juvenile periodontitis, but so these microbes are things like Canarella forsythia, one of my favorite species. Bacteroides, it used to be known as Bacteroides forsythus. So I did not discover these microbes, but I kicked myself in the butt because in 1996 I did discover a new bacterium. I isolated a bacterium that caused conjunctivitis in birds. And so I had the honor of actually naming that bacterium, but I didn't have the guts to name it after myself. So I named it after the bird that I isolated it from. But then I found out it's like some guy named Forsyth is naming bacteria after himself. I'm like, shoot, I should have done the same thing. But this guy Forsyth, and I'll tell you a little bit about him because as it turns out, I'll save that story. So these are microbes that are associated with adult periodontal disease. So this is at Harvard, those of you wanting to go to dental school should go to Harvard because the Forsyth Institute is part of Harvard. This is this is the leading oral microbiology research institute in the world, not coincidentally named Forsyth. So let me tell you the story of my great-great-grandfather. It wasn't him. Now, this guy's not related to me at all, but I always find it particularly cool. As a matter of fact, this is a picture of it that I took from a duck boat in the Charles River. This is the closest I've ever gotten to the Forsyth Institute. I always want to go in and introduce myself and tell them, my great-great-grandfather used to. But Forsyth was he was a great philanthropist. He was a dentist in the Boston area, and he used to donate large amounts of money to care for the dental health of homeless children and children of poor people. They were referred to as the Forsyth children, even though their names weren't Forsyth. And so this institute is named after him. Okay, therein ends the material for the next exam, right? So what we're going to cover from this point forward is going to be on, actually, the final exam. So remember that the final exam in this class is not truly a final exam. It's the last midterm. So the last midterm will be on the final period, which brings me to another thing that I keep meaning to put up something on Blackboard about. There is an error, some of you have picked up on this already, in the syllabus as to when the final is. When I wrote that final initially, I believed that this class was going to be at 10 a.m. because it used to be last year they moved me to 10 a.m. This year they moved me to 11. I changed the fact that the class is at 11, but the final, as I looked it up, is the 10 a.m. final. So actually, the molecular cell biology final is listed on our lecture. So that final date is wrong. So our actual final date is earlier. I think it's December 8th. I'm going to put something on Blackboard about it. Probably most of you have picked up on my error already. But during that final period, you will be tested on the material starting with viruses that we're about to talk about now forward. And then you can take a retake on one of the previous ones if you care to or need to. So let's just first do some general introductory material about viruses. And some of this stuff is probably material that you've picked up just in general living about viruses. So just by definition, what is a virus? So it's a genetic element capable of replication independent of the host chromosome, but not independent of the cell. These are obligate parasites. They cannot live outside of a cell. They can survive. They can maintain their integrity outside of the cell for some frequency, for some level of time, but they can't replicate themselves without the machinery of a host cell. So they're obligate intracellular parasitic entities. We won't even call them organisms because we're not going to get into the debate over whether these are living or not. They're not. So they do have a distinct extracellular form. This is important because I've been on this soapbox before when we talked about plasmids. So plasmids are not viruses, although I think they really are highly evolved viruses. Plasmids are not viruses because of this part of the definition of a virus. They do not have a distinct extracellular form. Viruses do. So that's how viruses spread from cell to cell. Conjugative plasmids, in my estimation, are viruses that have just evolved the ability to encode a sex pilus, a conjugal pilus, that they can pass through to another cell. It's just a highly evolved virus. But the other part of this definition that's really important is that viruses, these viral particles, are metabolically inert. Thank God. We don't have to go through any kind of central intermediary metabolism. There's no anabolic pathways going on within these viral particles. There's no TCA cycle, no glycolysis. Once they leave the cell, for the most part, they are inert. Occasionally, there's a proteolytic enzyme that's maybe still maturing some proteins, but for the most part, there's no metabolism going on within those extracellular particles, or virions. The virion itself, in terms of its morphology, can usually be classified into just a few different types of morphologies. Typically, they are icosahedral, those things that look like buckyballs. I'll show you an example of those. They can exist helically, or they can be complex. In other words, they're a combination of helices and icosahedrons. Unlike cellular life forms, they can use RNA as their genetic material. There is not a cellular form on life, or a cellular form of life remaining on Earth today that stores its genetic material as RNA. There are people who believe, and I happen to be one of them, that earliest forms of life on Earth probably did use RNA as their genetic material, because RNA is a wonderful molecule because it's not only informational, you can not only store genetic information in it, but it's also enzymatic. RNA has enzymatic activities many times. So many people believe that RNA used to be the cellular form of genetic material, but now no more. The only biological entities on Earth that use RNA as their genetic material are viruses. But not all viruses do that. Many viruses, like herpes viruses, things like that, are DNA viruses. For the most part, once we get past the bacteriophage portion of this class, that's the only DNA virus that we're going to talk about. My interest is more in RNA viruses, so we're going to use several examples of human pathogenic RNA viruses to explain virology. The other neat thing about these viral particles is that they contain only DNA or RNA, never both. So our cells have got obviously messenger RNA, transfer RNA, we've got the DNA, we've got small regulatory RNAs. Viral particles either have one or the other, not both. I don't really know why that is, but it's an interesting aspect to virology. And so, again, this is a microbiology class, and so we started small, and now we're getting even smaller. So we're going down now orders of magnitude in size. So I still don't like the fact that they use red blood cells in the comparison here, because red blood cells, remember, are one of the smallest human cells. So if you put in, a colonocyte from intestinal tract or something, it will be much larger than this. So this is a small eukaryotic cell. Even so, it shows you, here's the bacterium, E. coli, next to that. Here are the viruses down here. But if you blow them up and blow them up again, you can begin to see the size scale difference between these viruses and the bacteria and the red blood cell. So here's a bacterium, and here are the viruses. The largest viruses known are things like the pox viruses. This should be one word, smallpox virus, not a small pox virus. Smallpox virus is a pox virus, and these are large. These are actually large enough that if you have a good microscope, you can see them in the laboratory with your naked eye. Some people claim that. I've never been able to. Anything smaller than that, you have to use an electron micrograph or electron microscope for. So here are some other examples of viruses, and the nice thing is they show some of these different morphologies that I talked about. I'll talk a little bit about this one in a bit. Tobacco mosaic virus is the paradigmatic example of a helical virus. It's nothing more than a strand of RNA associated with multiple copies of the same protein, just attached to that RNA. This is one of the simplest biological infectious entities known. And it not only infects tobacco, but hopefully none of you smoke, but here's another reason not to smoke. These viruses in tobacco actually survive all of the processing that goes into making cigarettes. And this virus will also infect your tomato plants. So for those of you who are gardeners and smoke, you can potentially be killing your own tomato plants by touching them with your fingers that have touched tobacco. So I know everyone else will now quit, right? Hopefully they're under. I smoked for more than 20 years, so I'm one of those obnoxious people that likes to really preach at people not to smoke, because yeah. Anyway, moving on to other viruses. So here's an example of a complex virus morphology. So T4 bacteriophage has a helical section that's actually contractile, and then it has an icosahedral section here, which contains the DNA genome. But here's the stuff that really blows my mind. Here's poliovirus, responsible for epidemic polio, one of the scourges of the last century, that disease that hopefully we're getting rid of within the next decade worldwide. This little tiny virus, not much bigger than a ribosome, has enough genetic oomph to actually cause lethal diseases. This is just amazing that something this small could do this. But here's an icosahedral virus that is a naked virus. It's a naked virus because it doesn't have a lipid layer around it, such as the smallpox virus. What you're seeing here on the surface of this that makes it quite so large is a lipid envelope that was acquired from the previous cell that it infected. So now we're talking about very small viruses. And again, this is just an illustration of the different morphologies. So vaccinia virus, which is another pox virus, this is the virus that actually causes cowpox. This is where we get the term vaccine from, because Edward Jenner used cowpox virus, vaccinia virus, to artificially infect people to protect them from smallpox, because he noticed that milkmaids had very nice complexions back in the time because they weren't scarred by smallpox scars as many people would be. And so he reasoned that they must be something about milking cows must be protecting them. As it turned out, when he interviewed them, he realized that they had been infected with something that looked like smallpox, but it was much less severe and it tended to be limited to their hands and their arms. But it protected them from smallpox. So this is where we get the term vaccine from, is from this pox virus. But it's an envelope virus, kind of like this mumps virus here, which is an RNA virus. And then you have various complex viruses, and then you get down into these small icosahedral. These are the naked viruses because they don't have envelope. They don't have a lipid bilayer around them. This is that TMV, or tobacco mosaic virus, that I mentioned. That's the paradigm of a helical virus. And it's nothing more than an RNA genome with just many, many copies, 2,130 to be exact, copies of the same proteins just attached serially to this. If you extended that genome, they would just stick more protomers of that protein on there. Self-assembly. The protein just naturally binds to this RNA and forms this helix. Very, very simple virus. The envelope viruses are actually much more common among animal viruses. So we're going to start off, once we start talking about specifics of viruses, we're going to start with bacterial viruses, one in particular, lambda. And then we're going to move into animal viruses. Most bacterial viruses are naked viruses. They don't have lipid bilayers. So these envelopes are much more common among animal viruses. Like this is a mock-up of an influenza virus. So you have inside the 8 segments of the RNA genome of influenza bound to some proteins. But on the outside of this is host cell lipid bilayer. So it comes from the previous host that this virus infected. But these proteins that are on here are actually encoded by the virus genome. So we'll talk about influenza virus in great detail, because it's one of my favorite viruses not to be infected with, but it's one of my favorite viruses to use as an instructional tool. So the symmetry in a case like this is expressed not based on the outside, because these are squishy. You know, these can adapt, take on different shapes. But the symmetry of a virus like this is usually based on the nucleocapsid, which would be inside of this. The envelope almost always contains viral glycoproteins. So these are proteins that are encoded by the virus, not the host. And surprisingly, there are few host proteins that end up remaining in this. There's some, but not a lot. Most of the proteins that end up in this envelope before the virus leaves that cell are encoded by the virus. And they're going to help the virus infect the next cell, in this case in the respiratory tract, or perhaps the next person if this gets coughed on or sneezed on. So this envelope contains enzymes that are involved in the virulence or in the pathogenesis. In the case of influenza virus, one of the proteins that's shown here is the neuraminidase gene, which we'll talk about. This is a protein that helps release the virus from a cell so that it can affect another cell or get coughed out into another host, or to form attachments. So we'll talk about this other protein, the hemagglutinin protein. This is the protein encoded by the virus, now stuck into this lipid bilayer, that's going to help that virus bind to a specific cell in another host to initiate infection of influenza in that person. The complex viral symmetry is most elegantly seen in the bacteriophage. So we won't talk about T4. We're going to save time and just talk about lambda, but lambda will have some similar structures to this. So it not only has just an icosahedron head, but it's actually extended that head. So it's like putting a second floor on a ranch house, right? So because this virus has a large double-stranded DNA genome, it's actually added another layer of proteins to this icosahedron to fit more DNA. But this head has an icosahedral structure. The helical structure here shown is actually contractile. So viruses actually can contract like muscle. Now, this is not the same type of contraction that we talked about pili doing, or where you depolymerize a molecule. This is literally contracting because it twists on itself and just contracts that helix. When it does that, it takes the DNA from in here and drives it through the cell wall, the envelope of the bacterium, to infect, to initiate infection. So these viruses have partially helical morphology, partially icosahedral morphology. And the tail structure, this portion down here, as well as these pins, are really important for binding to the right bacterium. So the tips of these legs, the lunar lander type things, actually will bind to specific proteins on a particular bacterium. So this is what allows for this virus to infect a specific bacterium and not to necessarily infect virtually any other bacterium. And then these tail pins actually help to poke a hole in the cell membrane. So as these legs bind to specific proteins, they bind cooperatively. So if one here encounters the appropriate protein on the outer surface of a bacterium, it's going to increase the chances that this other leg will find an appropriate protein. And then once probably all six of these legs have found their specific protein, then that will initiate the infection. So this virus doesn't induce its contraction until really probably all six of those legs have bound to a specific protein. That way it doesn't shoot its DNA into the wrong host. Now, there's something very unusual about viruses in terms of being an infectious organism. They go through something that no other infectious organism goes through in the course of causing a disease. They go through something called the eclipse period. The eclipse period is illustrated really nicely in this graph of a bacteriophage infection of a bacterium in the laboratory. So on the y-axis here is the number of infectious virions present. You could do the same experiment with smallpox virus, you could do the same with poliovirus, and get the same effect. If you start with, let's say, one virus at time 0, about 5 minutes after that virus attaches, if you lyse open those cells, if you do everything you possibly can, you can't find that virus anymore. It's disappeared. It's not just protected someplace. It has literally been destroyed. It has come apart. There is nothing infectious any longer. And if you keep looking in that infection for 10 or 15 or 20 minutes, you'll see no viruses. And then all of a sudden, the virus resurrects itself. It's like a bad horror movie, right? So at 25 minutes, suddenly you can find lots of viruses, because what's happened is that the virus has been destroyed. Destroyed sounds like a passive process. The virus has actually come into its constituent parts during this eclipse period. So here, the virus has deconstructed itself and is creating many, many new copies of itself. So that period where there are no infectious microbes is never found in bacteria or parasites. It's only found among viruses. It's unique to the viruses. The reason that it's unique, this eclipse period, is because, exactly as I mentioned, the virus will deconstruct itself during the initial stage of infection. So here in this slide, we're illustrating 3 of the most common ways in which viruses gain access to a cell upon infection. In the case of something like poliovirus that we'll talk about, the capsid will bind to a specific receptor, and then its RNA genome, in the case of poliovirus, gets injected. Now, at this period, if you stop the experiment at that point and tried to find infectious poliovirus, they're gone. This RNA is now separated from the capsid, and it is never going back. It can't go back. Same thing here. This is an example of membrane fusion. So this is something like influenza virus would do, where the lipid bilayer can fuse Actually, I shouldn't use the example of influenza because that's really what's going on at the bottom. But some viruses, like for instance HIV, is probably the better example to talk about here. That lipid bilayer can fuse with the host membrane because in fact they're the same thing. They're both lipid bilayers. It's kind of like, think about two soap bubbles. If you put one soap bubble on top of another, it'll sit there for a minute, but eventually it gets subsumed by that bubble. The bubble becomes just slightly larger, and now there's one bubble. But that expels the genetic material of that virus or the nucleocapsid, which will then break down and release its RNA or DNA genome, depending on the virus, inside of that cell. But now the virus is gone. Same thing with influenza. In influenza, there's a kind of a phagocytosis event that goes on that then precipitates fusion of the lipid bilayer with the endosome. But in either case, you still get this eclipse period. You still get this period where there are no infectious viruses because the virus has come apart. Now, moving on and talking still some introductory material on viruses before we get into specific examples. You can look at viruses as either DNA viruses or RNA viruses. And this is where things get kind of complex because not only can viruses have either DNA or RNA as their genetic material, they can do it in ways that no other organism that we know of can. There are viruses that are single-stranded DNA viruses. That's their genome. They don't have a complementary strand to that DNA. But some are double-stranded DNA. And then with the weird RNA viruses that use their RNA as genetic material, they can also exist in either form, single-stranded RNA or double-stranded RNA. And then to make it even more complex, there are viruses like some hepatitis viruses and certainly the retroviruses that have the ability to toggle back and forth, the necessity to toggle back and forth between an RNA genome and a DNA form of that genome. They can be single-stranded RNA like the retroviruses or the hepadnaviruses. So these are viruses that cause at least one form of hepatitis. These are double-stranded DNA viruses, but they also have an RNA intermediate form. Really, so there's a tremendous degree of complexity. There's a lot of ways to get confused when you study virology. Because there's actually even yet another level of complexity among these viruses because they can not only be single-stranded RNA, that RNA can either be positive sense or negative sense, which makes it even more confusing. So just to introduce this terminology to you. So here's just an arbitrary piece of DNA of 9 bases that I just drew out. This is what double-stranded DNA would look like. If you were to transcribe that into messenger RNA, this is what the messenger RNA would look like, encoding 3 enzymes, methionine, histidine, glycine. A plus-stranded DNA virus, something like parvovirus is a single-stranded DNA virus. It would have DNA that looks like this, no second strand associated with it. You can have a negative strand DNA virus, and I don't know of an example of one of those, but the DNA that would encode the same amino acids would look like this. But if you translated that negative strand from 5' to 3', you come up with 3 very different amino acids. So that protein would not be functional. So a negative strand DNA virus has to first make a positive strand DNA, probably make that double-stranded DNA before it can make messenger RNA. If you had a positive strand RNA virus, this would be something like polio virus. Polio is a single-stranded, positive strand RNA virus. The RNA genome would exist exactly like this. So it could be translated directly. So if this is a poliovirus genome, as soon as it gets injected into the cytoplasm of a host cell, hopefully not yours or mine, it can be bound by ribosomes and translated directly into methionine, histidine, and glycine. But if a negative strand virus like influenza, so influenza is a single-stranded RNA virus, but it's antisense, it's a negative sense. If it needs to encode methionine, histidine, glycine, it's going to first have to make a positive strand copy of this RNA and then have that be translated. So very bizarre complexity. Do not ask why. We don't ask why in biology. It's one of those questions that, philosophers ask. People in religion ask why. In biology, we don't ask why, we just ask how. So it's a really complex series of genomes that these viruses can have. And they've actually been set up in a system not named for the city of Baltimore, but named for the Nobel Prize winning virologist who discovered retroviruses, David Baltimore. And on Friday, we'll go through the Baltimore system and talk a little bit more about viruses. See you on Friday.